On the right over here, you're seeing the changes in the follicle. Before we change slides, I want to emphasize that even as we go through all these changes in the follicle, notice that the cells at every level until we ovulate are primary oocytes. So even though we change the appearance of the follicular cells and all of that, it's still a primary oocyte. Internally, it's going through prophase and metaphase and anaphase until right before ovulation it completes telophase and we have 2 cells, right? But it's still a primary oocyte. All right, so when we look at the ovary, these cells all the way through here until ovulation are still, this is just the hour or so prior, are all primary oocytes. So let's look at those follicular changes. So move ahead to page 112 of your lecture notes. May I erase the board here? You guys have that down? Okay. One point that I want to clarify on any of these pictures or models of the ovaries is that the changes that are shown in these arrows are not correct in the implication that the egg is moving through the ovary. It's using the ovary to show the various stages, but this egg is going to ovulate at this point. The ovulation that takes place here originated with a follicle that looked like this right there. All right, so they don't migrate around. It's just showing the different stages, and it has to put it at a different place, or they'd all be on top of each other, unless they showed you successive drawings of ovaries. Okay, so it doesn't have to move through the ovaries, but stay right there. Now, I use some different terms, all right, than the textbook. So note that I never use the word primordial follicle. I start out with primary. Okay, so our egg is a primary oocyte. So just before ovulation. I'll keep writing it, reinforcing it. The follicular cells, when the baby is born, the baby girl is born, unless it develops receptors, and it's one of those 12 or 15 or so that start to move towards ovulation, are flat in a single layer. Simple squamous epithelium. These are the follicular cells. The ones that bind FSH, all right? At some point, they start to become called granulosa cells. I generally just stay with follicular because it's FSH that's binding to them. But if you see the word granulosa, it's just referring to follicular cells, all right? When the cells are single and flat, the follicular cells, we really don't notice. The theca interna cells are a little difficult to pick up. And so this is identified as a primary follicle. That's what the baby girl is born with. And that's what all the cells that are still, the eggs that are still in her ovaries when she reaches menopause, are still as primary follicles. All right, so it's only the ones that are preparing for ovulation that we start changing the name. So 10, 12, 15 of those cells start increasing the number of receptors that they have for FSH. What triggers that, we don't know. All right, this starts to happen about a year before actual ovulation, and in response, the cells round up and become cuboidal. So our primary oocyte is still present. However, our follicular cells round up, They're starting to get ready to synthesize estrogen. So what organelle do they need in the synthesis of estrogen? Do they need ribosomal ER or smooth ER? Is estrogen a protein hormone or a steroid hormone? Steroid, so they need smooth ER. But that's why they're getting bigger. So they're getting these organelles necessary for the production of estrogen. Okay? Conversion of androstenedione to estrogen. As soon as they round up, even though it's still a single layer, we now call this a secondary follicle. Okay? So that would be this layer here, secondary follicle. Now those follicular cells begin to undergo mitosis. By the way, what's the chromosomal pattern of a secondary of a follicular cell?
2N.
2N. Good. Remember, everything is 2N, except our secondary oocytes and spermatocytes and the cells that form after that. All of our body cells are 2N. Those cells start to multiply. Still a primary oocyte. And we still call it a secondary follicle. So it is still a secondary follicle. Now, if we consider that our follicular cells are starting to develop the equipment to convert androstenedione to estrogen, what do you think is happening to the level of estrogen as we get more of these cells? Estrogen levels are going up, all right? As estrogen levels go up, what's going to happen to the level of FSH? It's going to go down. So we're starting that, responding to that seesaw effect. So we're about this stage here, okay? We can start to see some of those theca interna cells. They're there, they're just hard to see, but now we can start to see them. They have to be there because they're making the androstenedione that the follicular cells are using to make the estrogen. So these are the cells binding LH. They're making androstenedione, which is moving into the follicular cells, which are making estrogen. All right? About this time, we also start to see the appearance, at least in the light microscope we can see it, of that protein coat, the zona pellucida. As we'll see on Thursday, the zona pellucida is going to prevent more than one sperm from penetrating and fertilizing the egg. All right, because it wouldn't be viable. Wouldn't be able to multiply and live. Again, still a secondary follicle, still a primary oocyte. Right? So this is about 2 months before this particular set of this egg is actually going to be ovulated. Now, as it continues to produce follicular cells and estrogen levels are building up, all right, estrogen starts to appear in clusters. Now we're talking about 2 or 3 months before it's ovulated. Okay, we have several of these cells doing this. So again, primary oocyte. Zona pellucida. Follicular cells, some of them are which are directly attached to the zona pellucida. And gaps appear. color do I want to use? Amongst the antral cells. Here we can keep a red pen. All right, we'll use purple. Gaps appear among the antral cells where fluid is collecting. The ciliary cells, and as soon as we see those gaps, even if they are scattered, this now allows us to classify this as an antral follicle. Because those gaps are antral. It's like the anteroom, a foyer, so it's a space. Okay. So this is classified as an antral follicle. Now, again, as we progress, those various spaces coalesce into one large space. It's still an antral follicle, which is farther along. Primary oocyte. I'm not going to make that zona pellucida quite as big. This single layer of follicular cells that are stuck to the zona pellucida are going to be ovulated with the egg. We now give them a name. The corona radiata. So they're right up there against the zona pellucida. The rest of the granulosal cells kind of form a cluster around that, a stalk that attaches it to the rest of the cells. And now we have, these are cells, not squiggly lines, but too lazy to form single cells. Still an antral follicle, okay? But one coalescing antral. Kind of like you see, like that one there. And then in the last month or so, only one of these really gets larger. Actually, really only one of them gets to this stage. And so, let me not forget my pica interna cells. Just don't want you to confuse it with the antra. So, this is going to get huge. And now we call it a mature antral follicle. Still an antral follicle, all right? But this largest one is called the mature antral follicle. This would expand out. So this would be the entire size of the antral. This wouldn't be here anymore. The antral would enlarge out to here, okay? Finally, there's so much fluid in there, under such pressure, that it ruptures. So a Graafian follicle is an old name I don't use. I just call it a mature antral follicle. And it finally ruptures and releases the egg and the zona pellucida and the cluster of cells. It's at this time that we see that first meiotic division, and we form the the first polar body, all right? So at ovulation, we would really have released our secondary oocyte, the first polar body, all of that surrounded by the zona pellucida, the corona radiata, and a bunch of other cells. That's what gets ovulated. Yes.
On a test, would you accept the circular follicle?
Yeah. You can show it in print somewhere. So looking in the light microscope, here, and you have these images in your lecture notes. Here's an egg. Here's its nucleus, the nucleolus, and flat follicular cells around it. right? Primary oocyte in a primary follicle. As soon as those round up, even if it's just a single layer, it is a secondary follicle. Okay? As soon as we start to see spaces, it's an antral follicle. Whether they're one space or one large space or many small spaces. So that was an antral follicle. This is an antral follicle. Here's the corona radiata. The white area here is the zona pellucida. And right out here are the theca interna cells. All right. Here's an actual ovulation. I think it was the first one captured on film. So what's being ovulated? Let me put my controls here. This is a pair of forceps or hemostat. Here's the ovary, which is about 1 inch to 1.5 inches long. You can kind of see the proportion of size. That's not the actual egg. It would be actually inside of that, and this is all the follicular cells surrounding it. Okay? It doesn't just pop out like popping a pimple. It kind of oozes out there. All right. This is painful for some women. There's a kind of a small cramping pain that occurs every month. Some women they may never feel that. Others may feel it a couple of times. And it's called Mittelschmerz. It's a German word for that kind of cramping pain that lasts maybe 20 minutes or so. Okay. So after question?
Yeah. So like the red,
the red is the surface of the ovary.
Of the mature antral
Right.
follicle?
Showing right here. Pushing out, and then the yellow would be this structure right here. Once the egg is ovulated, the cells that remain behind, the follicular cells and the theca interna cells, now start making primarily progesterone and turn very yellow in color because of the lipid, and so it's called a corpus luteum. Once that secretion stops, it forms a small residual body known as the corpus albicans or white body, and for every egg that was ovulated, there is a corpus albicans on the ovary. All right, so if we're looking at then our cycle here, kind of put together, we really have talked about 2 of these. We just need 3. We just need to put the 4th one in, because I want to finish up here by 3. So you have various components of these. It's not all together on one page, I don't think, but this is online. So here's what we've been looking at just now. Okay, the egg right here is a primary oocyte. Here it's a secondary oocyte. So we covered the changes of the egg. Here are the follicular changes. So before ovulation, this midline is ovulation. On a typical menstrual cycle, that's day 14. If you count the first day of bleeding as day 1, right here, that would be day 1. Day 14 is ovulation. And then 2 weeks later, menses starts again. That is only the true guarantee, is that 14 days after ovulation, give or take a day, menses will start. Once menses start, there's no guarantee when ovulation is going to occur. If a woman is anorexic, if she runs 50 miles a day, if she doesn't eat enough fat to make the steroid hormones, that could take forever. If she's under stress, if she's just starting her menstrual cycle as a teenager, it may be another 10 months before she ovulates again. All right? When she does, though, 2 weeks later, she'll have a menses. So it's not very good for birth control. All right, I ovulated 2 weeks ago. I shouldn't have had intercourse then. It's too late. If fertilization occurred, it's going to occur right here, about 7 to 9 days. We'll talk about that on Thursday.
So then you can't get pregnant when you're on your period. Because I heard people say, I got pregnant and I was on my period.
Breakthrough bleeding is yeah. If you're actually having your period, it's way past ovulation. But bleeding can occur. So it might have been that they actually ovulated earlier than they thought. And when they were bleeding, they were already pregnant. And it was just some mild loss of the stratum functionalis. Yes.
So the only follicle that's second degree is the mature.
The mature antral follicle, but only just prior to ovulation. Okay? So, what are the hormone changes that bring us to this point? We start out, really, menses is the end of the cycle. It's just easier to tag it at that time, all right? So, and you'll see, let me warn you ahead of time, you'll see charts in which in the early part of the menstrual cycle, FSH is higher than LH, and you'll see other charts where the reverse is true. Just to warn you about that, okay? We don't ever put these charts together because different units of measurement are used to measure them. So it's like saying, you know, I'm going to measure this in 5 cups and I'm going to measure this in ounces, you know, of weight. And it's just, you can't put them together, though you will see people who they do. So, primary follicle, FSH starts the follicular cells rounding up and preparing to synthesize estrogen. LH causes the theca cells to start making androstenedione. And now the follicular cells, there are sufficient numbers of them by the time we get to the secondary follicle, and sufficient numbers at the time of that month that have started way back before, that we see estrogen levels increasing. All right? Now, GnRH is going to be doing what? Low or high at this point?
Lower.
Seesaw, it's going to be lower. However, that's negative feedback, right? However, moderate high levels of estrogen actually cause a spike in GnRH, which causes a spike in LH and a smaller spike in FSH. That is positive feedback. So estrogen in lower to very high levels causes negative feedback, but here, positive. That surge in LH is responsible for the rupture of the mature follicle and ovulation. Within 24 hours of that surge, ovulation occurs. All right? Then we have an abrupt drop and a flat line again. All right? Look what's happening to progesterone. Now we start switching the hormone that's produced, and the theca interna cells forming the corpus luteum become more active, and progesterone becomes the primary hormone after ovulation. Estrogen is the primary hormone before, progesterone is the primary hormone after. We get a secondary rise of estrogen, but not nearly as high as progesterone. Okay? And that goes through till about the 3rd week of the cycle, day 21, 23, 24, in that range. Both estrogen and progesterone start to drop because our corpus luteum starts to fail. Not if the egg is fertilized. When the egg is fertilized, it makes HCG, the pregnancy hormone. All right, we'll talk about that on Thursday. If that occurs, it maintains elevated levels of progesterone. So why do we have menses? That leads us to the menstrual cycle. All right, so we come back to this chart. So here's the beginning of menses. Here's ovulation, all right, and then we see the preparation for the beginning of menses. So remember, at this point, our follicular cells are starting to increase. The estrogen levels are increasing. Notice what happens to the thickness of the stratum functionalis. It's getting thicker. Just like the duct cells in the breast multiply, and we get secretory cells, these cells multiply, the uterine glands increase in length. Now there's an error here. The uterine glands should actually start in the stratum basalis. Okay? So under the influence of estrogen, the glands increase, the connective tissue increases, and the overall thickness of the uterus increases, the endometrium. After ovulation, remember our primary hormone is progesterone. Progesterone's role is to increase the blood supply. So the blood vessels in the endometrium grow very rapidly, and they branch because the blood vessels have receptors for progesterone. Remember, progesterone levels drop. Corpus luteum is only maintained for about 8 or 9 days. And when progesterone levels drop, those blood vessels constrict. What happens to tissue if it doesn't get blood supply? It dies. So as that stratum functionalis tissue is dying, it loses its attachment to the stratum basalis, and we get menses. So estrogen is the growth and thickness of connective tissue, glands, and epithelium, which is maintained after ovulation. Progesterone is the increased blood supply to support that tissue. Ever heard of the abortion drug RU486?
Yeah.
All right, it binds to progesterone receptors and blocks progesterone from binding, but it doesn't have the same effect on the cell. So it's essentially removing progesterone from the system. And this is the result. The tissue dies and separates. If there's an embryo embedded in here, that is lost with the stratum functionalis. Okay? As we'll see on Thursday when a baby is born, the placenta separates. The afterbirth is the placenta and the same stratum functionalis that you lose during menses. Only the baby's already out. Okay? So this separation is that line. So this kind of puts all of that together, and you have this slide on D2L, so you don't have it all as one piece. So hormonal cycle affects both the follicular ovarian cycle and the endometrial or uterine cycle. Okay? And we'll talk about how this continues with fertilization on Thursday. So we'll stop here. I think I'll go ahead and do what I did this morning and let you have a break and come back and study whatever you want with the models. Remember, the quiz is male structures, muscles, glands, just the same thing that was available on last Thursday. But now there will be a slide of the seminiferous histology and one other slide of the male organs. Okay, no female histology. And then a couple of slides of female organs, ligaments, parts of the uterus, and the nerves. So pudendal, sacrosplanchnic, or pelvic splanchnic. All right, so let's take this, let's aim to take the exam. We finished about the same time, and we not the exam. I took the quiz this morning at quarter after. All right, so let's look. I'll check with you at 4, and then we'll have a quiz sometime between 4:10 and 4:15. We'll have it at 4 if you're ready, but sometime between 4 and 4:15, okay? And then we'll do the clay model. And while you're doing that, I'll pass out the quizzes. Go ahead and take a break, and I'll do that when you guys get back, or while you're take a break now and grab you guys when you get back.
Oh, I haven't finished that yet.
What's your outside?
Or to also provide, I have hormonal migraine. So they often come at ovulation or at the beginning of menstruation. It's effect on blood vessels. I would almost think about 6 months and then I quit. And I don't know if that was the cause, but yeah, it started, it took a while, 2 or 3 years, and then I noticed it was happening every other cycle.
So it was.
And it kind of changed, so it was happening more often, but not as severe. I wasn't in bed for 3 days, kind of thing. So now I get a really bad one every February. It's just kind of, yeah. I have a really bad one on November side. That's really painful like this. But yeah, after that, they last like a week to 3 days, but they're not, they don't throw you to bed, kind of feel, just a change. Yeah, my doctor said, we should have Minnesota, maybe you won't get them as often. Is it all? Rub it off.
I mean, the first one. I don't know. Because you can't really tell on the, that looks like the chromosomes are already divided, so.
Okay. Do you take it when you have it, or until it continues to make?
Like this one, I do not understand what that was.
I'll bring you some. Okay, thanks. Yes. So the quiz on the male is all of this, but also this which we didn't cover last time, and the histology. Okay, but there'll be, yeah, I asked some of this already, but I'll ask some of the slice. And then on the female, it won't be, so guys, on the female, it won't be any of all the parts of the egg and the follicles and stuff like that. I won't ask any questions about follicles. It's all the parts of the uterus or the ligaments or parts of the vagina or the erectile tissue, stuff like that, but not inside the ovary and not histology. So it won't be any of the parts of the ovary. So starting with oviduct, here, down through all of this. Actually, I don't have a mammary gland on it, and then none of this. Okay. Sure. So this won't be on it. Oviduct. No mammary glands. And no histology. Well, pudendal nerve, same as for the male. And then
For female, it starts with the oviduct,
okay.
and it goes until this, until the pudendal nerve. And then the male, the same thing, everything.
I'm going to put price on the back end. I think it might be a good right there.
Professor?
Yes.
And the male is whole thing, right? Front and back?
Yes. One slide on the nerves, one slide on the histology, and then the other slide, any of that stuff that was up for grabs last week. No.
Male and female on here.
Is that a capsule bird cell? There's no capsule around it? I was wondering how that
She said probably say what? She said it could also be Oogenesis.
I'm picturing the same map, same. Not that.